Can you hear me? Can you hear me? Can you hear me? Can you hear me? Okay, I'm speaking. Can people hear me? 1, 2, 3, testing this microphone. Thank you very much. Hello, everyone.
Hello, 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 hello.
Welcome to this session. My name is Daniel. I'm the director of the Soil Ecology Laboratory, and this is Adam. He's the bio-inoculum engineer at the lab. First things first, we have to apologize. You were expecting Elaine here, and she really wanted to make this up until the very last minute, but she double booked herself with a flight to see her family, and this would have been 5 in the morning for her, and she would have been doing this from the airport. So common sense took over. So very much apologies from her and from ourselves. And to get to this session, we've got 45 minutes, so we're going to try and be as practical as we can. But to give you a bit of background, I was Elaine's first online student here in the UK back in 2017, so I was mentored by her directly. And then in 2019, I quit my job to start the lab. And shortly after, Adam joined me, and we've been trying to develop what Elaine teaches. If you go back to 2015 to the Oxford Real Farming Conference, you will find that her presentation in 2015 is still the most watched video from all of the videos on the conference. So it's understandable that people are interested in what she has to say. And then last year, me, Elaine, and Glyn did a presentation online as well, where we're trying to basically show that we're actually now moving forward. We're trying to actually make this a reality in the UK. And when I first started, basically, I saw that there's 2 issues with getting her message implemented and actually making this a mainstream thing. And the 2 things is access to this biology, and the second thing is reliable set of data of trials that were performed with this biology against controls and against the conventional systems. And so the lab has been set up basically to find the truth, to find those 2 things, so we can give farmers an alternative. Because we're all for banning things, but if you just ban things, it leaves a hole, and something has to fill it. And in the past, when you banned something, you just basically had another chemical come in, another chemical come in. So our mission is basically to do this broad scale. So we're actually working towards allowing farmers on the broad scale, whether they have 50 or 100 or 2,000 hectares. We want to help every farmer at every level to be able to implement this easily. So the first issue is actually having, I'm going to stand up so people can see me in the back, actually having a good source of biology. And so I actually reached out to the green waste industry and spent probably a year just looking at samples down the microscope of compost from the green waste industry. And this is actually what Adam is going to be presenting now. So first we're going to see what you actually see under the microscope when you look at that kind of compost. So the logic behind actually going to the green waste industry is that they process close to 2 million tons of green waste per year. And so if you are trying to actually push this as a mainstream thing, you actually have to have a good supply of material. So we're going to talk about all of the other work that we do individually with farmers so that you can do things on your own. But I just want to say that this is what we've set up. We actually want to push this forward. We want to get that message of Elaine's into all of the farmers' hands so they can actually just use it and they can make up their own mind on their own land. So I went to the green waste industry in 2018, and I actually was invited as their keynote speaker 3 times, and I was in their magazine, industry magazine, 3-page article about how can they actually have a better product that they can actually sell, because oftentimes they actually have an issue with selling the product. So what Adam is doing right now is preparing the sample by shaking it in the exact same manner every time. So it has to be consistent. If you're doing these samples, you have to be consistent. So he's doing this, he's been doing this for 3 years now. His arm is just basically just falling off there. But you have to be consistent with the process that you're choosing. So One of the things that we're going to show you, actually, there's a little playlist that you can email me. I can send you the link to that playlist. There's a playlist that I created on YouTube that takes old videos of Elaine's that somebody commissioned her to do on how to prepare samples and put it on the microscope. It's free. It's not part of her course that she needs to pay a lot of money for, but it's the start of it. So if you're doing things on a budget, just get that playlist from me, and you've got a little starter that you can actually already start on the microscope. This is that playlist, and it basically goes through choosing the microscope setup, how to take the sample, and then how to prepare it. It doesn't show you how to identify the organisms, I'm sorry. But at least you've got to start how to work on the microscope, okay? So we're going to share that with you.
Welcome to get in touch with us, and we'll teach you the way.
Yes. We're actually very open. So one of the things that we talked about with Elaine very early on is to set up hubs around the world. And the way the school went in one direction wasn't allowing Elaine to do that, but I actually took that, and we're very open. You can come to us, you can spend a day with us, you can call us. We've got a Zoom call every Wednesday that you're welcome to drop in at any week. And anybody who's actually going through the course with Elaine, we've got another Zoom call on Thursdays. So it's all free. Come and talk to us. So the thing with the composting industry, I did make a lot of acquaintances there, and the person who's actually the head of the organics recycling group is on our advisory board, because we need that regulation at some point as well if you want to scale this up. So we need to be in line with the regulations. And so the people in there, they're open for new ideas, and they want to connect. They want to make a good product. At the moment, they're thinking of that product only as, we've got this material coming in, we get paid a gate fee, we need to clear it as quickly as possible so we can get new material and get paid for that gate fee. So they're not thinking about creating the value yet. So what we're trying to do is we're trying to be the leaders to show them that they can actually do that. And so we're actually strategically located on a farm which actually operates a green waste site. And they process, I think they're licensed to 5,000 tons a year. So it's not a big site. It's not like Veolia or Envar, but we're actually talking with one of those as well. So we've specifically located ourselves because we wanted to have access to the same input materials that they've got, and then basically try and experiment. And we've experimented with all sorts of materials, and we can talk about that too. This is the website, soilecologylab.co.uk. Give us a shout, and we're going to show you something interesting on this website in a minute. This is another website, soilsmiths.co.uk. You can actually, I'll talk about that later. But we've located ourselves on this farm with green waste. It's a conventional farm. So fertilizers, fungicides, glyphosate, everything's gone. Everything's going as the agronomist prescribes. And then in 2021, we did a trial on spring barley on 0.5 hectare. Last year, we did a trial on winter wheat on 5 hectares. Now we're actually scaling back the trials, and we're looking to actually look deeper into the processes, and I'll talk about that in a minute. So what Adam is doing right now is actually just preparing a slide. And so he did the shaking, and then he dropped a drop of the extract onto the slide, and then covered it with a cover slip. We're going to go through that again. So you're going to see that again. And that's basically the process. So it's, whilst I was talking, he just prepared that. So we're going to go through that again so you can see it. But I just want to mention that this is not a difficult process to learn. The difficulty is actually in just the fact that you have to each time, set up the microscope and prepare, and you have to actually do it. So I find that the most difficult thing. Once I sit down in front of a microscope, I can spend a few hours, and that's not a problem. And then your eyes go absolutely blind from the LED light. Have you got something there now?
Nearly there.
Nearly there. Okay, great. So the first sample, yes, we're looking at...
This is green waste, straight from the windrow.
So Adam is using a 10 times objective right now, and pretty much the only two objectives that he's using is a 10 times objective and a 40 times objective, and then you've got the ocular at 10 times magnification. So your total magnification on here is 100 times. And what you can see here is basically, there's pieces of organic matter. Oh my, there's a nematode, that's amazing. And there's pieces of organic matter, and you can see that this is actually not really very well decomposed. You can actually see the cell structure here. So all of this stuff is basically relatively fresh. And then these little pieces, you know, they could be potentially even some minerals that are just basically mixed in with the lighter one especially. You can't obviously, this is, hold on one second. Mm-hmm. You've got a grain of sand here, for instance. So the thing is that in this sort of compost, the majority of stuff that you see is basically, there's a lot of bacteria. All the little dots are bacteria. There's going to be some organic matter. There's going to be probably some mineral fraction. And the nematode was quite lucky, actually. Adam, do you have some more to add to this?
Yeah, here as well. It might have been on top of the cover slip, but it looks like a piece of microplastic.
There we go. That is actually a fiber. You can see the color of it. And yeah, you get to see some of those too. This was, if you were here at the previous conversation, this was about creating a peat-free alternative for horticulture. This is something that the green waste industry actually is trying to solve. But they don't really have yet the incentive to do so. And so the problem with plastic is actually on the council side. The council collects the material, it goes into the material, and then they shred it all. And I know literally 2 companies that have taken the step of removing the plastic before the shredding. And they literally have to do it by hand. There's no other way at the moment to actually get rid of that. So this guy is, Adam, over to you.
Yeah, so this guy's a nematode, most likely a bacterial-feeding nematode. We can tell that by looking at the morphology, the mouth parts, the esophagus. And this is actually pretty lucky to see, and it's most likely because the green waste windrow is active. If it was finished, often it goes very hydrophobic. And I bought some hydrophobic material, but it was just too hydrophobic to make the sample itself. So this shows you how bad that stuff gets.
Which compost is this?
This is from Robert's site.
This is Robert's, yeah.
And you've got 2 nematodes, right? But this is, what you're looking at is bacterial ecology. So you're looking at bacteria. This is a bacterially dominated substrate. And it contains ecology based off that bacterial ecology. So just scroll through and you can get a kind of sense of what this material looks like, how much aggregation is in there, how much mineral component is in there. These clues give you a good insight into where this material is at, especially when you start to look at the development of material as it ages, as it matures. Often, like was mentioned in the previous talk, this material has excesses or deficiencies in the chemistry that make it quite unusable. You know, and as peat gets banned, we're going to look at peat replacements. At this stage, this isn't really very good for plant growth, depending on which plant you're growing, but most plants will be compromised nutritionally by this material, as well as held back by the lack of ecology, both on the lower trophic stages and the higher trophic ecology that's built off that. So if we just scroll around, we might see some interesting stuff. You might see a flagellate. It doesn't look like there's many around. We're just going to see bacterial-feeding nematodes. We're going to see the mineral components. We're going to see these aggregates. And here you can see like some filaments, some clear filaments maybe holding some of the aggregation together. If we looked at this stuff with DNA analysis, it would show stuff. It would show fungi, it would show bacteria, and it would show higher trophic grazers, but it wouldn't give us any idea of abundance or of the active biomass. And that's why microscopy is still super important, trying to understand that abundance, trying to understand what players are actually present and active in the ecological scene of this compost or soil or substrate that we might want to develop.
And it's important to actually note that all of those tables of interpretation of fungal to bacterial biomass ratio that you may have heard of succession basically, that the higher the fungal component in relation to the bacteria, the further you're going towards an old growth forest from like an desert pretty much, nothing, rocky outcrop, to an old-growth forest. And the thing is that all of those tables of interpretation that 1:1 is pasture and 10:1 are trees and shrubs and trees for fruit production, and then 1,000:1 is an old-growth forest. Those were done with a microscope. And so there are other methods that you can assess fungal to bacterial biomass ratios. And when you do that, do it with the caveat that you need to do it against some other table of interpretation. And the same comes with DNA analysis. Now, this is an interesting story in itself. Elaine always keeps talking about, don't worry about looking at species level, just get the groups right, leave that to your grandchildren so they have work to do for their PhDs. And Elaine turned 70 in August, and I said to her, I said, I could be a grandchild, right? So basically, that's why we're actually looking at DNA analysis now. And next week, actually, we're starting a trial in a grow room, completely controlled, with a soil from the outside, which had all the chemicals and whatnot. And we're going to be trying to grow wheat, but we're going to be measuring nutrient analysis in the soil and in the extracts. We're going to be measuring microscopy of the soil microbes and the extracts. We're going to be doing the DNA analysis of the substrate and the extracts of compost. And we're also going to be doing sap analysis to find that end point. Because although all of this stuff is great, it's one thing that's most important to think about is that microbes are a pump. The microbes basically are supposed to be the mechanism that delivers everything. So we need to have that thing working. So what we're trying to do is we're trying to look at it deeper so that at the end of all of this, at the end of all of that exercise, you can go into confidence with confidence into this and say, like, okay, well, if I make my Johnson soup bioreactor, if I keep it for 12 months, if I make sure it doesn't dry out, if I make sure that several other things are taken into account, maybe the starting materials, by the month 12, I've got a good chance that actually I have the material I can experiment with. And if you have that website actually up, we have actually been receiving samples from farmers of their compost. So if you go to the soil biology testing, this one, yeah, and if you scroll down. So we've actually put them into a bit of a top-tier leaderboard, and we've marked them against fungal to bacterial biomass ratio. But what we've been getting is actually all of these things that read on the side is what type of compost it was. And majority of it is actually Johnson soup bioreactor, Johnson soup bioreactor, Johnson soup. So the thing is that the Johnson soup bioreactor actually has become this easy entry for farmers to try this out. There's no mucking about with turning compost. There's no worries about that. You just basically put that cage and leave it. And you don't have to follow even the notion of watering it because we're not in Arizona. Just leave it outside. It's the UK. It rains. So we've been getting various results, and you can see that what farmers are getting from those Johnson soups, they're definitely getting bacteria, they're definitely getting protozoa. They don't always get the fungi, but a lot of them do. And then they don't always get the nematodes. So we've been actually trying to bump the numbers of those two groups, the fungal and the bacterial feeding nematodes, to actually get to a more complete product. There's some interesting ones here. There's a found material. You can't see the numbers again. But the owner of that stands in the back over there, Niels. And he basically found 14-year-old wood chip. And the reason I'm mentioning this is that time is on your side. That if basically you give enough time to this material in the right conditions, you will get the diversity and the volumes and the numbers of microbes that you can actually then use. And in the previous conversation here, Tolly was talking about how he actually keeps his wood chips for 2 years before he uses it. So if you want a simple system to start with, go for a Johnson soup bioreactor and forget about it for 2 years. And then basically every few months, whenever you have time, just make new piles. And then learn to use the microscope to assess it, or send us the sample, or some other specialists who are doing the sampling, and just basically then you're sure that you've got the kind of material that you need.
And so what happens over that period of time of the maturation of compost is that the excesses or the deficiencies get corrected, and the compost moves towards more ideal soil chemistry, which makes it better to grow higher terrestrial plants, right? And some of those excesses from the chemistry move into microbial biomass. So particularly the fungal groups. You'll get more successional fungal development as the earlier kind of primary decomposers, they degrade the parent material, and the metabolites from those groups cascade through the fungal digestion in a process of terrestrial humification of the material. So now what we're going to look at is that succession. So it's taken us a long time to develop this product and try and make it relatively uniform, and we're pretty proud of it. It regularly shows levels 5 to 10 times the minimum requirements outlaid by Dr. Laningham and the Soil Food Web School. And it's a refined compost extract, so we can deliver it without the weight of the compost itself or any kind of aqueous solution that might be carried in. We'll take them at the end, if that's cool.
Just to help understand, is this green waste starting off sterile from a high-temperature compost?
Completely sterile is not probably the right word, but yeah, as you hit those higher temperature ranges, you really you start to compromise the diversity that it carries for pathogens and for beneficials.
If you think about it, actually, if you look into, for instance, Gourmet Mushroom book, Growing by Paul Stamets, he actually mentions composting, high-temperature composting, as a means to prepare the material for the colonization of the gourmet mushrooms that he wants to grow. So what you're doing is actually you're just reducing the competition so that your preferred organism can actually then grow. The whole point of thermal compost is to reduce pathogen load. Now, all that means is that basically there's only a small number of pathogens so that the other guys can flourish and they can just out-crowd them, sometimes eat them, but basically just the good guys are just taking over the pile. So the temperature is a main prerequisite, and it comes from the food industry. So it basically means that if you keep the material at a certain temperature for long enough, then you get a low pathogen load. Now here's the thing. Elaine will tell you, keep it at 55 degrees or above for 3 days. Keep it at 65 degrees or above for 2 days. 75, 1 day is enough, right? If you look at the actual literature of reducing pathogen load in the food industry, you keep it at 65 degrees for 1 hour. So this is enough. You don't really, you can actually manipulate it quite quickly. And so, but what the green waste industry does, they read the PAS 100 standard. And the PAS 100 standard is a piece of, it's just a standard that comes from the conversation between the industry and DEFRA. And they say, you are required to go through a serialization process. And then look at the guidelines. It's not a requirement, it's the guidelines. And the guidelines say, you will get a low pathogen load if you keep it at 75 or above. So that's what they do. That's all they do. When you start reaching 80, 82 degrees, 84, even the thermophilic bacteria start dying. The guys that are responsible for raising that temperature, they actually start dying off as well. And the other thing that happens in that high temperature is that some of the simpler sugars that are used by the microbes to eat, they actually get caramelized. So they're not accessible then. So then it takes a while, for the more complex organisms then afterwards to get to them. What we've been actually able to do, we've taken the material from the farm, this material, and we've been able to demonstrate that you can actually turn it around. We first started with a 3-liter pot. We added our extract from our better compost. Then we took it to a 40-liter pot. We added our extract. It proliferated. Then we put a whole cage. We call it the Bob Sonsu because Robert is the owner of the farm. And so basically that's his compost with our microbes. They proliferated. And then we took a big pile that was put out to the field that was waiting for it. And we came back after, 4 months? Yeah. Something like that. And we went to the back of the pile where we didn't put the microbes. And they all went through it. So the thing is that you can take bad compost and make it good with time and with proper inoculation.
But you can also optimize that process, so you can dramatically speed it up. So we've seen good compost made in as little as 4 months. So yeah, what we're looking at now, you can obviously see some differences. You can see some differences in the amount of microaggregation, this kind of micronization of the parent material that the microbes do. And you can see some more microbes on the scene, or at least I can, and hopefully by the end of this, you'll be able to recognize them too.
Johnson-Su bioreactors is a form of Johnson-Su bioreactor. It's a form of static compost. It's a glorified leaf box. That's all it is.
And that's something you add to the basic green waste?
No, it's something that you can put together so that you can have your own compost of great quality. Thank you. And here, Adam is actually finding some fungal strands for you.
So it would have been it would have been near enough impossible to find this in the green waste compost we first looked at. And that's because this process of fungal digestion I was talking about previously hasn't been given time to be completed. So these guys are the tertiary decomposing fungi, the ones that are more happy in soil. So if you think of soil as a three-dimensional thing, we have a mulch layer, we have the interface between the mulch layer and the true soil, and then we have the mineral soil. The primary decomposers will be happy up top in the mulch layer. Think King Stropharia, think types of mushrooms that produce fruiting structures. They'll break down that mulch, and it will become smaller and smaller particles, and the lignin, the cellulose will be broken down. The metabolites that are excreted by those forms of fungi go on to feed later successional forms of fungi, and that's kind of what we're looking at here. This even diameter, evenly septated, so those cross walls are very even. Often they'll show a kind of dark color, whether it's dark brown or black sometimes, or even purples and reds, like brick red colors. They show that these hyphae are sequestering materials. They're setting up shop, so to speak. They're less likely to be opportunistic pathogens because of that characteristic of setting up structure.
And this is what actually Elaine talks about in her training. She classifies good guys and bad guys kind of with a variable of time, because to really sort of go with her definition of a good guy, you need that fungal filament, that hyphae, to actually have, as Adam was saying, to have a bit of meat on it. Because what the fungi will do as it grows, it will take anything that it will find, it will attach it to the side of the hyphae on the inside. And it will be there for storage, it will be there for sequestration, it will be there for, oh, I found a toxin, I'll just cut it up here and I'll just put it there so it doesn't bother me because I'm over there, trying to find new nutrients. So it will build that hyphae. And then as we shake the sample, as we sort of prepare it, the hyphae will break up, and it will break up actually in very predictable manner, in a 90-degree manner. Now, the other thing, on the flip side, what Elaine will say is that most likely the pathogens, opportunists, they don't live in this long cycle. They will basically just take all the nutrients they can and run. And so their hyphae is less developed. It's thin-walled. It's often frothy on the inside. And often the septate, the division between the cells, is at different distances. And so that general definition is what she will say, that these guys potentially are pathogens. Now, I like to put in a caveat because we've looked deeper into this now, and I say that any freshly growing fungal hyphae will look like a pathogen to you. And so this is something that if you have a pile that's been sitting for a while, then you know that you're looking for those well-developed ones just to be there. You want them to be there. But if you were to take King Stropharia, wine cup mushroom, or if you were to take oyster mushroom of any kind and took the fresh hyphae under the microscope, you will think it's a pathogen because it looks like one in the definition of Elaine's. And so there's been numerous conversations about how to identify it, but still, we're looking for that Elaine's definition of building that structure so that you know that they've been there for a while, and you know that they've been out-competing stuff for a while. The other thing to actually note in this thing is that all of those aggregates, you can't tell what it was anymore. This is fully decomposed. This is, humics, fulvics, just basically really complex jumble of carbon molecules that basically have been processed by the microbes over an extended period of time. So there's and Adam, I think you're trying to show us some other organism as well.
Yeah, so in shot, you could maybe point to it, Daniel.
Yep.
We have a testate amoeba.
This guy here.
Wouldn't have seen them in the green waste compost. Super valuable organism in cycling bacterial biomass. Much like the bacterial feeding nematodes or any of the bacterial ecology, they open up the packages of nutrition that have been obtained from the organic matter or from the mineral component of the compost, and they make them plant available. This is what we call nutrient cycling. This is the process of mineralization. This is how wild plants forage for nutrition.
And this is actually the basis of what Elaine teaches, because she was part of the original team back in 1985. There's an ecological monograph, the main paper that she goes off, in terms of the basis of what she teaches, is that they were the first to actually demonstrate that you can have bacteria, you can have fungi in the soil, but if you don't have the nutrient cyclers, the bacteria and the fungi act like, as Adam was saying, micro-fertilizer bags. And somebody has to come in, with a bag opener and actually open them up. And that's the role of protozoa and nematodes. And this is why we do these assays, so we can count them. We can say, per 1 milliliter of your compost or your soil, you've got this many nutrient cyclers. And there are some calculations, if you're really nerdy, that you can actually then take those numbers and then you can translate that into the actual amount of N, the actual amount of other nutrients that the plant could be getting per day, for instance. There's now many papers, that I have seen that actually demonstrate that with the protozoa, you actually get mineralization rates at a much higher level than if you just had the bacteria, the fungi. And so that's why making your own compost, this is another one that Adam wants to talk about.
Yes, another form of testate amoeba. It's known as an arcella. And we'll see these pop up as the material successes towards fungal development. You notice how they're kind of a similar color to the environment they're in. They're like this fulvic color. So maybe their tests are made from that material, a product of that environment. But again, an amoeba that will cycle nutrients held in bacterial biomass.
You also see to the left top, you actually also see some fungal spores. And usually the better ones, if you just see simple round spores, you're happy. You're not seeing anything that is worrisome. So there's actually charts of spores that you can find online, and you can educate yourself. There's also a very good group on Facebook called Fungal Microscopy. If I don't know something, I just ask them. They usually tell me. And you can identify it that way.
So what we do at the lab is go through these samples. We manually count these microbes, and we enter them into a spreadsheet. And from that, we extrapolate a kind of estimation of active biomass or active numbers of microbes within the material. And that's a pretty powerful tool, because you guys can all do that at home. You can get a microscope, you can use a spreadsheet to extrapolate that information, that data, and it gives you an idea of where in succession is your compost, how much bacteria in relation to how much fungi. And that gives you an idea of how much nitrate is being cycled compared to how much ammonium, dependent on the ratio between the bacteria and fungi and the presence of the hyotrophic grazers. It also gives you a really quick way to understand whether an input you use, like say an Ascophyllum nodosum seaweed extract that says boosts microbes in the soil on the packet, does it actually do that? If I get 10 millivit per liter and I apply it to this compost, what do I see? So it gives you a way of kind of quantifying for yourself whether an input you're using is beneficial, if it's destabilizing to the microbial ecology, or if it just outright starts to kill everything off. And that's obviously very useful for you guys as growers, when you're looking to save pennies. So we've got one there. If you want to show everyone, it's It's 40 ml of water with 10 ml in. It was hydrophobic, so it's had some time to absorb the moisture. And you could just probably go through again for everyone how to extract and.
That's yeah. I'll leave that with you. You're the expert here.
Okay. Hang on. Did we get a flagellate go? Why? Or is it? Oh, it's going to be hard to see a flagellate on this level anyway.
Yeah.
Maybe the top of the screen now. You can see movement, but it's a bit out of focus.
Yeah, flagellate is going to be a bit of a challenge to show on this screen. But the whole point of this is that we have used this methodology to then to really perfect the way we actually work with the compost. So it is just like Elaine designed, basically a simple way to assess if you have the right microbes. And we know now that there is a lot of potential in this. As I was saying earlier, we did a test on spring barley. We beat the control by 40% with just 2 sprays, overhead sprays. And they were not amazing, I can tell you that. On the winter wheat, we did 4.9 tons of wheat per hectare with 0 extra nitrogen. So that means that we beat the average in the USA and Canada, so we're happy with that for now. We'd like to get to the UK average of about 8 tons. We had our clients in Scotland do a trial on pasture with our biology, and they pretty much almost matched the biomass of 50 kilograms of nitrogen with 1 application of our biology. And the application was 1.5 milliliter of this per square meter. That's it. And then we had some other people try it with hemp, good successes. And then also, there's a very good trial now that's been repeated on radishes. So if you show the Saltsmith's website.
Where am I going?
On the top. Top. There we go. And if you scroll down, these are tomatoes in Playsand, by the way, from B&Q. If you scroll down. So a friend of ours, Silke, she's also a student of Elaine's. She basically took some of our goop and then she just planted radishes in Saturn compost. So Saturn seed compost bought from online or from a store. And she got a 55% increase in the weight of radishes without any extra fertilizer. So that was great. We were very excited that she did that. This was last year. But then I thought, it makes no sense unless we repeat it. And so we repeated it towards the end of last year, and we had a 22% overall increase, 27% average increase in the weight of radish, and the largest radish difference of our biology against the control was 88%. So the thing is that what we've been able to do now is go towards that goal of making this material so that first of all people can experiment and then start gathering the data experiments. So we're almost out of time, so I want to leave some time for questions. But all I just wanted to say is that we're available for you to talk to us, and we can help you with your own composting so you can make your own material, or you can have some of our material and you can experiment with that. So at the moment we're not at scale, so it's pretty expensive, sorry. But we're actually looking for investment right now so we can scale up this thing and make it a reasonable price at scale. We're aiming for a price that's going to be competitive with actually artificial fertilizers. And that was before the price hikes. So we're here. We're available for you to ask any questions. Every Wednesday there's a meeting for farmers. You can drop in and ask us questions. We don't hold back. And then there is material that you can buy on the website if you want to experiment before you get your own material after 2 years. So
the material is also really good for anyone going through the soil food web courses to have some kind of reference material to look at the microbes, you know, and practice your identification, practice your counting. This material goes beyond your local woodland. I've been to a 10,000-year-old woodland, and you don't see microbes as dense as what we call the goop, right? So for anyone that's doing the soil food course, you're welcome to come up and take some home, look at it, play with it, apply it to plants, see what you find. And just to go quickly back to the screen, this is more indicative of what you actually see in green waste compost, that hydrophobic material. Yeah, so you can see zero aggregation, right? Why isn't it moving? It's kind of frozen.
What we also get is a lot of soil samples as well. And any soil sample from an agricultural field that's been plowed, fertilized, fungicide used, all of these things, pretty much all that we see is the mineral fraction in bacteria. There's nothing else. And the interesting bit is that there is a lot of bacteria, meaning these micro-fertilizer bags are there, but there's nothing to open them. And so we attribute at least partially the success of that spring barley, for instance, and the winter wheat to the fact that the nutrients are in the soil, most likely, locked up in the bacteria, but we've actually produced the flagellates, the protozoa, the nematodes that can go out and actually release it to the plants. So there's a vast amount of nutrients in the soil already, but they just have to be released. Questions? Yes?
I think we're only going to have time for a couple of quick questions because we have got quite a tight turnaround, but I'm sure you'll be outside afterwards.
Yep.
If people want to chat with you. So.
Absolutely.
There's a man over there.
Hi. I was just wondering how you store your goop and what its shelf life is.
Very short. It's a living product, so we advise to use it within a week. This is based on and use it basically straight away or keep it in the fridge for a week, no more than that. Now we have done that, so we know that up until the week we basically lose the nematodes, and then for 4 weeks the fungi keep, no problem, and then at week 5 we start to smell things. But for 4 weeks there's no mold, there's nothing.
So it's a product that will be made fresh, shipped to you, you use it straight away, ideally. Yeah. And that's how it's going to operate at scale too.
Yeah.
Any more for any more?
Oh, there's one over there.
I was just wondering about on some of the slides, say the amoebas, for example.
Yep.
Have they died now, or are they not very mobile? Or?
No, what you are seeing is testate amoebae. They sleep a lot. So they'll create that shell around them, and they do it quite rapidly, actually. And they basically just sit in that shell until basically they say, okay, I'm going to go out and do something. When they go out, they kind of look like a slug. They just put out this pseudopod, and they'll just drag that shell behind them, and they'll encapsulate anything else, and then they'll just drag behind it.
Yeah, because the extraction process is quite disruptive, they'll often grab a lot of debris and pull it into the stoma. So what we see looks like an inactive nematode, but amoeba. But if you look within the test, you'll see the organelles. You can kind of identify that it's actually living and active, just not.
Sometimes you actually see just the empty shell, and you can actually see that it's just a shell, nothing in it, and then you don't count that.
Yeah.
Thank you very much for coming.